Okay, let's jump right into this explainer and take a grand tour of the endoplasmic reticulum, or as we usually call it, the ER. If you really want to understand how a eukaryotic cell actually survives and functions on a day-to-day basis, you absolutely have to understand this microscopic master manufacturing network. We're not just looking at the ER as some static biological structure today. We're going to look at it as a massive, highly efficient factory complex, one that's totally responsible for protein synthesis, lipid production, and some seriously ruthless quality control. So our itinerary for this explainer covers 5 clear departments. We'll start with the master factory, move into the assembly line of the rough ER, then the chemical hub of the smooth ER. After that, we'll check out quality control via ER stress, and finally wrap up with disease and biotech breakthroughs. All right, Section 1, the cell's master factory. Now, the ER isn't just some tiny floating blob hanging out in the cytoplasm. It is a massive, interconnected network made up of these flattened membrane sacs called cisternae, along with this really complex web of tubules. But honestly, the most crucial architectural feature here is its location. The ER membrane is physically, literally continuous with the outer nuclear membrane. It's an absolutely brilliant design. It means there is a direct line of communication between the cell's command center, the nucleus, and the factory floor. This allows for instant, seamless coordination between genetic instructions and physical manufacturing. And this factory is fundamentally divided into 2 major regions that, despite being connected to each other, have very different structures and jobs. On one side, we have the rough ER. It gets its name because its surface is just studded with ribosomes, which handle all the heavy lifting of protein building and folding. On the other side, we have the smooth ER. No ribosomes here, which gives it a sleek appearance, and it basically serves as a highly specialized chemical processing hub, entirely focused on lipid synthesis and detoxification. Let's dive into Section 2, rough ER, the protein assembly line. The ribosomes attached to the rough ER are hard at work synthesizing very specific types of proteins, specifically those entering the secretory pathway. We're not talking about your everyday proteins for the cell's own internal housekeeping. These are premium products destined for crucial external or structural roles. They're manufactured to be secreted outside the cell, embedded into the plasma membrane, or used in lysosomes. Think about it. The life-saving insulin your body needs to regulate blood sugar? The antibodies your immune system relies on to fight off infections? Or the digestive enzymes breaking down your lunch right now? All of these are roaming right off the rough ER's assembly line. But, simply linking amino acids together to build the protein just isn't enough. A protein only actually works if it's folded into a very specific 3-dimensional shape. And that's exactly where molecular chaperones come in. Enter BIP, or binding immunoglobulin protein. You can kind of think of BIP as our diligent line supervisor stationed inside the ER lumen. It physically assists these newly synthesized raw proteins, making sure they fold into their correct 3D structures. Proper folding is absolutely non-negotiable for protein function. So let's see how this builds into a complete logistics pathway. Once a protein is assembled and perfectly folded, it doesn't just sit around waiting. We can trace its exact route. It starts at the ribosome, moves into the rough ER for processing, and then gets packaged into a transport vesicle. That vesicle acts exactly like a microscopic delivery truck, ferrying the finished protein out of the ER and over to the Golgi apparatus for final modifications and shipping to its ultimate destination. It's just a seamless, highly coordinated pathway. Moving right along to Section 3, smooth ER, the chemical hub. Now, this chemical hub handles a highly diverse portfolio that's literally critical for your body's survival. The smooth ER is the primary manufacturing site for essential cellular lipids, including phospholipids and cholesterol. You need those to build cell membranes and organelle membranes. But it doesn't stop there. In certain specialized cells, the smooth ER is entirely responsible for producing steroid hormones, like testosterone, estrogen, and cortisol. On top of all that, it plays a vital role in metabolizing carbohydrates. If you've ever wondered how your body handles foreign compounds or medication, just look at your liver cells. The smooth ER in the liver is completely packed with a family of enzymes known as cytochrome P450. These enzymes are essentially the biological equivalent of a hazardous waste treatment facility. They constantly metabolize drugs, alcohol, and foreign toxins, breaking them down and modifying them to make these substances much easier for your body to safely eliminate. And the smooth ER gets even more specialized depending on the cell it lives in. For instance, the ER acts as a major intracellular storage unit for calcium ions. In your muscle cells, this takes the form of a highly specialized smooth ER called the sarcoplasmic reticulum. Every single time you move a muscle, whether you're sprinting down the street or literally just blinking your eyes, it's happening because the sarcoplasmic reticulum is rapidly releasing its stored calcium to trigger that muscle contraction. It's amazing. Which brings us to Section 4, ER stress and quality control. So, what happens when the factory floor backs up, or a product is hopelessly defective? It's a critical, high-stakes question for the cell. What actually happens when defective, misfolded proteins start piling up on the assembly line? A cell simply cannot allow defective proteins to enter the secretory pathway. Doing so would be disastrous for the whole organism. It needs a way to catch these errors immediately. The cell answers this massive problem with ERAD, which stands for ER-associated degradation. This is a brilliant, highly efficient disposal mechanism. When a protein is just hopelessly misfolded, ERAD identifies it, physically yanks it out of the endoplasmic reticulum, and tags it with a molecule called ubiquinin. This tag acts as a biological trash label, basically directing the defective protein to the proteasome, where it's completely degraded and destroyed. This is what keeps the factory floor clear and running smoothly. However, sometimes the backlog is just too massive. If unfolded or misfolded proteins accumulate too rapidly, the cell enters a state called ER stress. And this triggers a full-blown emergency protocol known as the Unfolded Protein Response, or UPR. The UPR is monitored by three major stress sensors: PERK, IRE1, and ATF6. These sensors will take drastic measures to save the cell. They'll temporarily halt new protein production to stop the jam from getting worse, and they'll rapidly increase the factory's folding capacity. The goal here is to restore homeostasis. But if the stress is too severe and the cell just can't recover, the UPR will actually force the cell to self-destruct. It triggers apoptosis, programmed cell death, to protect the rest of the body. Brutal, but necessary. Finally, let's look at Section 5, Disease and Biotech Breakthroughs. Because when the ER's quality control fails, or when mutations overwhelm these systems, the consequences for human health are severe. Protein misfolding and ER stress are directly linked to devastating neurodegenerative diseases, like Alzheimer's, Parkinson's, and Huntington's disease. Furthermore, prolonged ER stress can damage the insulin-producing beta cells in your pancreas, which directly contributes to diabetes. And in a much darker twist, cancer cells frequently hijack and exploit these very ER stress survival pathways to keep themselves alive under toxic, unfavorable conditions. But conversely, scientists are now heavily exploiting this cellular megafactory for our benefit in biotechnology. Because the ER is just so incredibly good at producing properly folded proteins, researchers use eukaryotic expression systems to mass-produce life-saving recombinant proteins. By leveraging the ER's natural machinery, we're able to manufacture complex therapeutic antibodies, synthetic hormones, crucial enzymes, and the very modern vaccines that we rely on in medicine today. And this frontier is expanding rapidly. Today, researchers are combining AI-assisted protein folding models with synthetic biology to engineer these quality control systems from the ground up. We are now targeting specific ER stress pathways as a completely new avenue for drug discovery. By understanding exactly how the ER manages stress and folding, we're engineering cells to improve protein production yields, entirely paving the way for the next generation of medical breakthroughs for metabolic and neurodegenerative disorders. To wrap up our tour, I've got a quick revision table for you here from the source material. It breaks down everything perfectly. You can clearly see how the rough ER dominates protein synthesis and vesicular transport, while the smooth ER handles lipid synthesis, detoxification, and calcium storage. And of course, overseeing it all are the vital quality control mechanisms, ERAD, and the UPR. Feel free to pause for a second to lock in these core concepts from the explainer. So the crucial point to take away from this explainer is perfectly captured in this beautifully simple summary quote right from our source text. The ER truly is the master manufacturing and quality control network of the cell. Knowing how deeply this intricate, microscopic system impacts everything from your basic muscle movements to complex diseases, how might we one day re-engineer our own cellular factories to cure the incurable? That is the exciting question driving the future of biology. Thanks for joining me on this deep dive.